Okay, again, anaerobic respiration in yeast causes الblue dye اللي هو methylene blue to become colorless. When the anaerobic respiration works, the blue dye turns to colorless. Okay. The student wants to investigate the effect of temperature on the rate of respiration. If I'm investigating the effect of temperature, what will I do? I will change the temperature and then I will check the respiration of the yeast. So you will underline the temperature and write changed variable. Okay. So step number 1, 7 cubic centimeter of yeast suspension was put into a test tube labeled warm. The test tube was then placed into a beaker of warm water. And the temperature of the beaker was 45 degrees Celsius. Okay. In step 2, 7 cubic centimeter of yeast suspension was placed in a test tube called cool, and it was placed in a beaker of cold water, 20 degrees Celsius. طيب, since you placed 7 cubic centimeter of yeast suspension هنا with 7 cubic centimeter of yeast suspension here, so you put equal volumes of yeast in both beakers. يبقى what type of variable is this? Controlled variable. This is controlled variable. The volume of the yeast is something you put the same in both beakers. Okay. After 3 minutes, the student added 5 drops of methylene blue to the yeast suspension in each test tube. برضو, if you put the same number of drops, yes, this is an error, but before stating the error, if you put the same number of drops, 5 here and 5 here. So what type of variable is this?
Controlled.
Controlled variable. Controlled variable since you put equal number of drops in both. طيب. Is using the drops accurate? زي ما
لا عشان ممكن ياخد different volume أو حاجة.
Yes, the drops are not the same size. Okay, so يبقى it is a controlled variable. يعني ممكن أكتب over 5 that this is a controlled variable since you put 5 drops in each. However, putting drops is an error. Why is it an error? Because the size of the drops might be different in each test tube. Okay. The yeast suspension became blue. When you add the methylene blue, the color will be blue. Okay.
Is this another error that he added the drops in both the test tubes?
No, this is the results that he's measuring. Didn't we say when the anaerobic respiration works, the blue becomes colorless. So what he did, he put 1 test tube in hot water, 1 test tube in cold water, and he wants to see which will work faster. How will I see this? The one that will do anaerobic respiration faster, it will turn to colorless faster. So this is the results that you're going to see. Okay. He then put layer of oil over the yeast to maintain anaerobic respiration, to prevent the oxygen from entering, so that the yeast can do the anaerobic respiration. Okay. A stop clock was started. He observed the warm and the cool test tubes. When he no longer sees the blue color, he records the time. There are 2 errors in this step. One error was observing the 2 test tubes at the same time. This is not an accurate way because you will not able to record the time accurately for both test tubes. The other error was the color. We know that judging the color by the eye is difficult. It is difficult to know when exactly the end point where the color changed. And we said to improve this, we should use a colorimeter. So here we could write, this is an error, which is using the 2 test tubes at the same time. And number 2, observing the color using your eyes. This is another error. Okay. Figure 1.2 shows the stopwatch. He wants you to draw a table of results. But take care convert the time to seconds. Okay, what do I do? Which variable, if I'm going to draw a table, which variable do I put on the left side?
هنحط control.
Which variable do it changed
temperature.
variable? Yes, the changed variable is on the left side. طب what did we change in this experiment? We go back كده. We changed the temperature. We put one test tube in hot water, one test tube in cold water. طيب, what are the results that you are measuring? What are the results? What were we recording for results? We were recording the time taken for the blue color to disappear. Okay, so this is the how you should draw the table. Here is the temperature. One was warm, one was cool. You can even add another column to be more accurate, and you can write the actual temperatures. يعني ممكن نكتب هنا, we can write here beaker. This is the warm and this is the cool. And we can write here temperature because he gave me the temperature / degrees Celsius. Hina it was 45 and then it was 20. Type. And then please write the full title. Time taken for blue color to disappear. Okay. And then you will record the time that he gave you, but you have to change it to seconds. 3 times 3 times 60 plus 53. 9 times 60 plus 25 to convert the numbers into seconds. Okay? State a conclusion for the results. What is the conclusion? As temperature increases, time taken for methylene blue to change colorless decrease. This is the conclusion. Okay. The student used a thermometer to measure the temperatures of the warm water and the cool water at the end of the investigation. At the end of the investigation, يعني this beaker, it was supposed to be at 45 degrees Celsius. This beaker was supposed to be at 20 degrees Celsius. Type. If you measured the temperature at the end of the investigation, what was the temperature بقى?
Miss, I didn't understand what is the
Again, I will explain it again. 45 degrees Celsius, this was the original temperature in the warm beaker. 20 degrees Celsius was the original temperature in the cool water. But we said if you left the water bath on the table, it will cool down. It will not remain at the same temperature. And this is what happened. If you check the final temperature, you will find that here it was 35 degrees Celsius and here it was 15 degrees Celsius. Okay? So you should write هنا temperatures. At the start, it was 45. Do not write the units again inside the table. At the end, it was 35. هنا at the start, it was 20. At the end, it was 15 degrees Celsius. Okay. This is, of course, is not accurate. It's not accurate because the temperature has to remain the same throughout the whole experiment in order to know the effect. Okay, so here what did we say? In order to maintain the temperature the same, you have to use a thermostatically controlled water bath so that the temperature remains the same throughout the experiment. Okay, identify the variable that was changed. We already marked it at the beginning. We said that he was investigating the effect of temperature. So he is going to change the temperature to check its effect. Identify 2 variables that were kept constant.
concentration of yeast suspension.
Yes, we also identified these at the beginning. We said that the volume, not concentration, it's cubic centimeter. So this is the volume of yeast suspension. It was constant. Also, the number of drops of methylene blue. We put 5 drops here, 5 drops here. So this was also constant. What were the two variables kept constant? And the volume of yeast and number of drops. Okay. Identify one possible source of error in step 6 and suggest an improvement. We said 2 errors in step 6. One of them was observing both test tubes at the same time. This is not accurate. What should I do? You should do each test tube separately. The other one was observing
Judging the color.
Yes, it is difficult to determine the end point when the color changed. So you should use a colorimeter. Okay, يبقى difficult to determine the end point by eyes. What is the improvement? Use colorimeter. Okay. The data you have recorded may indicate that there is a source of error with the method used in this investigation. هنا he's asking me about this source of error بقى. The data that you recorded in the temperature was something at the start and at the end it cooled down. What is the error here? What is the error in this part بقى? What shall I say? So the error will be temperature is not maintained. It will cool down with time and this is not accurate. The improvement will be use thermostatically controlled water bath. Okay, anaerobic respiration in yeast produces a gas. What is the name of indicator which could be used to show that the gas is carbon dioxide? And what is
Lime
the
water.
expected result? The lime water, yes,
Lime water
Turns milky.
and
turns
it
milky.
turns milky or it turns cloudy. Okay. There was also the hydrogen carbonate indicator we took before. It turns yellow with the carbon dioxide. Hydrogen carbonate is red. When the carbon dioxide passes through it, it becomes yellow. Type. Question D. Design an experiment. Anaerobic respiration in yeast also produces ethanol. In high concentrations of ethanol, can slow down the rate of respiration. Ethanol, if it's high, it slows down the respiration. Plan an investigation to determine the effect of different concentrations of ethanol on the rate of respiration. The first step is the replica. How many test tubes will you use? And we said the more the better. Type.
4 or more.
I can use,
5.
yes,
5.
use 5 test tubes. Test tubes. Number 2, what do I do? For each test tube, what will you put the same بقى? As we said before, same volume of yeast suspension. He also told me this above. He put 7 cubic centimeter of yeast. Same temperature. I do not need to change the temperature in this investigation. Why? Because I am determining the effect of different concentrations. So this is the changed variable here. The changed variable is the different concentrations. Everything else should be the same. 1 second.
مش ينفع سؤال؟
1 second. Same pH. Okay. So all of these should be the same. I will also put the same volume of methylene blue. Same volume of methylene blue dye. Methylene blue dye. Type. These are the constant variables. What will I change? He told you that he wants to investigate the effect of different concentrations. يبقى put different concentration of ethanol in each test tube. This is the changed variable. After the replica, which is how many test tubes you will use, you will put the constant variables or the fixed variables, and then you put what did you change. Number 4, you put the results, not the actual results. I just need to know how will you measure your results. For this experiment, you will do the same as he did before. What was the results he was measuring? He was measuring the time taken for the blue color of the methylene dye
to be
to
colored.
disappear. Yes, so I will also do the same. Measure time taken for blue color to disappear. Number 5, you will put an improvement to the investigation, and then a safety precaution, and then the last step is repeat experiment and take average. Okay, since we said it has to be at the same temperature, constant temperature, يبقى we use thermostatically controlled water bath. Thermostatically controlled water bath, which has an electric heater that prevents the water bath from cooling down. Thermostatically controlled water bath. Number 6, safety precaution. Of course, you have to wear goggles. Number 7, repeat experiment and take average. Repeat experiment and take average. So these are the 7 steps for designing an experiment. Question 2, it's a photograph showing 4 seed heads from a poppy plant. Number 1, draw a large diagram of the seed head labeled A. How will I draw this? You should take care from a few things. Number 1, how many sticks? You should count the sticks. 1, 2, 3, 4, 5, 6, 7, 8, 9. You have 9 rods, so you have to draw 9. And then between the rods, there is like an outline of a circle between the rods. And then around the rods, there is something like a flower. Okay? After you draw the flower, you put the outer circle, which is هنا. There is a large part of the circle showing, while here the top flower is covering this something that looks like the flower, it is covering the seed. So you have to draw this, shift it a little bit upwards, not totally in the middle of the circle. Okay? It will be something that looks like this. But even here, you have برضو to I should have put the circle a little bit downwards. Make it a little bit larger from here. And erase this. Okay. This means we have 9 sticks. We have a circle in between. We have a flower-like shape around the sticks. And we have a circle where this flower is shifted a bit upwards. It's not in the center. Your lines has to be clear. Yani do not do connections like this between the lines. And your drawing has to be large. It has to be larger than the image. Okay? After the drawing, it will give you a magnification question. Measure the length of PQ, include the unit. Using your ruler, you will measure the length of PQ in centimeters, and then you will write centimeter. Or if you want to put it in millimeter, then you will have to multiply by 10 and put the number that you measured using your ruler and next to it millimeter. Calculate the actual size of the head using the formula. You have the length of the line, you have the magnification. Where is the magnification? It's always under the image. And you need the actual. طيب, to make it easier, you can put the triangle. Magnification equals image over actual. So if you want to get the actual, you will put the image size over the magnification. The image size is what you measured here. The magnification is what is below the image. Type. The image size will be around 2.6 centimeters, which is equal to 26 millimeters. What about the magnification? It is 1.6. I think under the image it was 1.6. Yes. So I will put 26 divided 1.6. It will give me the actual length, okay? Which is, sorry, did you measure the line PQ?
Yes,
it was,
it was 2.4.
Okay, it's okay. 2.4 is fine. According to the printing scale, it will be يعني still okay. طيب 2.6 divided 1.6. What's the answer? Okay, 26 divided 1.6, it will give me 16.24, but take care, he wants it to the nearest whole number, so we will round up. This will be 16 and include the unit. Since you took the image using the millimeters, يبقى الأكtual will also be in millimeters. Okay? طيب. A student investigated the effect of pH on the germination of seeds. He changed the pH and he wants to see the seeds that germinated. The student planted 25 seeds for each pH value. Okay, he gave you the number of seeds that germinated in each dish. Calculate the percentage of seeds that germinated at pH 8. So here, he did not ask for percentage change. He asked for a percentage, just the normal percentage. And he told you that the dish had 25 seeds, 15 seeds from the 25 germinated. So I will put 15 over 25 times 100 to calculate the percentage. It will be 60%. Okay. Plot a line graph to show the effect of pH on the percentage of seeds that germinated. Line graph to show the effect of pH on the percentage of seeds that germinate. So step number 1, the pH is the factor you changed. You put it on the x-axis. The percentage of seeds, which is the factor you measured, you put it on the y-axis. Step number 1 is putting the labels on both axes, like this. You have to put the name. Hina, we put pH. Hina, we put seeds germinated / percentage. Okay. Step number 2, put the scales. If you don't have numbers before 4, you don't have to put the numbers on the scale. You can start with 4. Okay? So here I started with 3. Okay. 3, 4, 5, 6, 7, 8, 9. But what if you started from 4 and you put 4, 5, 6, 7, 8, 9, 10? Bardo is correct. For the percentage, you have here, what was the number? It was 60. The smallest number is 40. The largest number was 96. So, what is the scale that I will put? What's the scale that I should put here? السكيل هيبقى ايه بقى؟ برضو since the smallest number is 40, you do not have to start from the 0. Here, I started from 20, 40, 60, 80, 100. The most important thing is that from the highest point to the lowest point in your graph, you should cover more than 50% of the graph area. Also, from the left to the right, you should cover more than 50% of the graph area. This is the rules for the graph to draw it. You then start to plot the points بقى. At 4, it was 80%. At 5, it was 92. At 6, 96. You start plotting your points. You should put the points as X or a dot in a circle. After you put the points, you connect the points, join point to point using your ruler. And this is how the graph should look like. Describe the effect of pH on the percentage of seeds that germinated shown on your graph. So if I want to describe, what will I say? As the pH increases, the percentage of seeds germinated increase and then decrease. The second point is you should use data quote. For example, I could tell him that the highest percentage of seeds germinated was 96% at pH 6. 96% at pH 6. Okay? So these are the 2 points for the description. The student wanted to obtain a more accurate value for the optimum pH. Can I say that this is the optimum pH? Can I be sure that this is the optimum pH for the seeds? I cannot make sure that pH 6 is the optimum because I didn't try anything around it. I did not try 6.5. I did not try 5.8. If you tried these pHs, it might give you higher percentage of seeds. So how can I be more accurate? What shall I tell him? I will tell him repeat experiment at smaller intervals between pH 5 and 7. Repeat the experiment again, but put smaller ranges. يعني مثلا 5, 5.5, 6, 6.5, 7. You have to put a smaller interval. Okay. The last question, during germination, the starch within the seeds is broken down to sugar. Describe how you could test a sample of seeds to find if it contains reducing sugar. Okay, so the answer will be add Benedict solution, heat, very important, in water bath. If sugar is present, color will be orange red. Will be red or orange red. Okay, and that's it.